Okay. Okay. Hopefully that has given time for anybody who is joining to join us today. Welcome to another episode of ALS Clinical Trials Unboxed, brought to you by ALS Society of Canada. My name is David Taylor. I am the Chief Scientific Officer of ALS Canada, and we are joined here by Dr. Lauren Kett. Dr. Lauren Kett is the head and medical director in clinical development at Amylyx. And many of you will know Amylyx, who have been the company that has had Albrioza here in Canada, and now is been doubling down over the past several years on looking into discoveries for potential new therapeutics and experimental therapeutics in ALS. And here to discuss AMX 114, a new experimental therapeutic coming to clinical trials here in Canada, is Dr. Kett. And I will pass it off to you. Welcome, and the floor is yours.
Wonderful. Thanks so much, Dave, and the broader team as well. I'm so excited to be here today and able to kind of be here talking about AMX 114. As Dave mentioned, this is, we're excited to kind of be back with a new therapeutic that we are looking to investigate in individuals who have sporadic ALS. Wanted to kind of come and give an update on the scientific rationale as well as our study design as we're moving this into the clinic. Just 1 minute about me. So my name is Lauren Kett. As Dave mentioned, I'm a medical director here in our clinical development group at Amylyx Pharmaceuticals. My background, I'm actually a neurologist and a neuromuscular physician. I saw individuals with ALS in the ALS clinic at Mass General Hospital before jumping into conducting clinical trials in people with neuromuscular diseases like ALS. And today, my goal is to talk to you about AMX 114, which, as we mentioned, is an antisense oligonucleotide targeting calpain-2. I will also be talking about the LUMINA study, which is our first in human study investigating AMX 114. So at the start, I do have a brief disclaimer, which is that AMX 114 is an investigational product and has not been determined to be safe or effective by any healthcare authority, for example, Health Canada, the FDA, et cetera. This presentation is really intended to provide scientific information about AMX 114. So here is the agenda of what I would like to cover today. So the first thing is I want to provide the rationale for why we are interested in studying calpain-2 in ALS. Next, I'm going to kind of go into a little bit of details about AMX 114, which is the drug that we will be studying here, kind of our rationale for why we chose to go after this target, as well as why we chose to use an antisense oligonucleotide to do it, and then some of our preclinical data for why we're so excited about this compound in individuals with ALS. Finally, I'm going to talk about our LUMINA study design, which is our first in human study looking at AMX 114 in individuals with ALS. So we'll cover the study design as well as our current status. And then at the end of the time, we'll go into kind of any questions that you all may have about the study or the compound or anything else that I talk about. So for my first topic, why are we studying calpain-2 in ALS? So the science behind this actually goes back really decades. One of the things that we have learned over the last 20 or 30 years is that axonal degeneration is actually one of the earliest processes kind of leading to ALS disease pathogenesis. So axons are the ways that our nerve cells communicate with each other in the brain and the spinal cord. And it is also the way that nerve cells communicate with the muscles that are moving in your arms and your legs. So shown here in the picture in this kind of red purpley color, this is the nerve cell. In ALS, this nerve cell typically sits kind of in the spinal cord. And then it sends this really long kind of electrical wire, this axon, all the way out, either the arm or the leg to make connections with the muscle. And so when the nerve cell says, move, it sends that signal down the axon and then tells the muscle cell to kind of contract, generating the movement. And so These axons are the things that really allow neurons to talk to each other as well as communicate with the muscles throughout the body. In ALS, what we see is that one of the very early things that happens is there is a disruption in the ability of the nerve cells to kind of just talk to the muscle. And that disruption happens because of axonal degeneration. So axonal degeneration is the breakdown and the disintegration of all of the different building blocks of these axons. And this is an event that tends to happen very early in ALS, as well as kind of in other related neurodegenerative disorders as well. Calpain-2, which is really the protein that AMX 114 and we are especially interested in, is something that contributes very early to axonal degeneration. So calpain-2 is a protease. This means is that when calpain-2 gets activated or kind of turned on, it runs around and it cleaves other proteins, also known as substrates. So calpain-2 kind of gets activated by very high levels of calcium inside the cell, and then it has the function of going around and kind of cleaving or cutting up other proteins inside the axon. When calpain-2 gets activated or overactivated, that leads to axonal degeneration. So that leads to that disruption and disintegration of the motor axons communicating with the muscles. And so as a result, what we see in ALS and other neurodegenerative disorders is that the overactivation of calpain-2 leads to axonal degeneration, leads to the reduced integrity of a bunch of different cellular structures, and then that leads to kind of the degenerative process. So we think that this is something that happens very early in the disease pathogenesis. There are a few kind of specific reasons for why we're targeting calpain-2 in ALS. So the first reason is that we know that calpain-2 levels are elevated in people with ALS. This is based on a few different studies where they took either cerebrospinal fluid, kind of the fluid that is surrounding the brain and the spinal cord, and looked to actually measure calpain-2 levels in people with ALS. They also looked at muscle biopsies in individuals with ALS, and in both of those situations saw that calpain-2 levels were much higher in people with ALS compared to people who didn't have ALS. So that kind of was one of the first signals that it seems like there is something going on with calpain-2 that might be linked to the disease pathogenesis in ALS. The second thing is that we know that calpain-2 acts directly on key proteins in ALS. I mentioned that calpain-2 is a protease, so when it gets activated, it kind of goes around and cleaves other proteins. Well, some of those proteins that it cleaves includes things like neurofilaments, which are structural proteins in those axons that have been, highly, highly studied in ALS. We also know that calpain-2 cleaves proteins like TDP-43, which is another key protein in ALS pathogenesis that tends to accumulate inside neurons. So we know that calpain-2 levels are high. We also know that it is linked to kind of other key disease proteins in ALS. Additionally, there have been studies that have been done in ALS mouse models that show that if you inhibit calpain-2, there is actually benefit in terms of in terms of motor neuron survival, as well as in terms of muscle function. And then the last thing, and I will go into this a little bit more on the next slide, we also know that if you knock down levels of calpain-2 using AMX 114, that we have improvement in motor neuron health as well as in motor neuron survival. So because of all of these reasons combined, we thought that this was a very interesting target that we could try to affect. If we can reduce levels of calpain-2, we think that we will have a benefit in terms of ALS disease progression. So next I want to shift gears very slightly and talk about AMX 114, which is the drug that we are studying in our upcoming clinical trial. So AMX 114 is an antisense oligonucleotide, or an ASO, targeting calpain-2. So over here on the right side of the screen, I have kind of a brief schematic of what an antisense oligonucleotide is. For people who are familiar with the drug tofersen or Qalsody, that is a different ASO targeting the SOD1 mRNA. So, what normally happens inside a cell or a neuron is you have a stretch of DNA. In this case, the stretch of DNA, has the gene for calpain 2 encoded on it. What the cell does is when it's trying to make calpain 2, that DNA is then converted into a messenger RNA or an mRNA. And this is a single strand of an mRNA that the cell then uses to convert the mRNA into a protein. So in this case, you have, the calpain 2 stretch of the DNA gets transcribed into the calpain 2 mRNA that then gets converted into the calpain 2 protein. When we add an antisense oligonucleotide like AMX 114, the antisense oligonucleotide very selectively and specifically binds just to the stretch of the mRNA that is important for calpain 2. And as a result of that binding, this then kind of targets that mRNA to be destroyed within the cell. And so the end result then is that you have no or decreased synthesis of the calpain 2 protein. So by using the ASO, we're kind of very selectively and specifically able to reduce the levels of calpain 2 within the cell. This was very appealing to us as a way of kind of targeting calpain 2 for a few different reasons. One, we know that we're able to kind of very highly reduce the amount of calpain 2 that is expressed within the cell. The second thing is that we're really able to kind of selectively and specifically target calpain 2 instead of other related proteins. Like there are other calpains that are in the body. There is also kind of the cell's natural calpain inhibitor called calpistatin. And so by using an antisense oligonucleotide, we're only targeting calpain 2. We're not kind of going after those other related proteins. And then the second thing is that we're able to, really lower the levels of the calpain 2 mRNA, and as a result, lower the amounts of the functional calpain 2 protein. Also, by using an antisense oligonucleotide like this, the effects tend to last longer. So, you give one dose of an antisense oligonucleotide, and the effects are to reduce levels of calpain 2 for a much longer period of time. We have studied AMX 114 in a number of different cell models, looking at ALS. So this is my slide to kind of walk through the data that we have from cell models of ALS to show that AMX 114 has a benefit. So the first types of experiments that we conducted were to take motor neurons in a dish and then essentially injure them in a few different ways and see if knocking down the amount of calpain 2 is protective against that injury. So in our first experiment, experiment A, which is all the way on the left side of the screen, we grew motor neurons in a cell dish, and then we treated some of them with AMX 114 to reduce the amount of calpain 2, and then we treated some neurons with a control or a placebo. So they have normal amounts of calpain 2. We then take those two different dishes of motor neurons and we expose them to different compounds that are toxic to the nervous system. So in this case, we used compounds like vincristine or rotenone or colchicine. And then we measured the injury to the motor neuron axons by measuring how much neurofilament light chain was released into the media. And what we saw is that motor neurons that had been treated with any of these kind of toxic compounds typically release high levels of the neurofilament light because as a result of that injury, they have that axonal degeneration and they release the neurofilament light into the media. When we pre-treat those motor neurons with AMX 114 and reduce the levels of calpain 2, there is less axonal injury and there is less neurofilament light chain in the media. So that's the first type of experiment we did. In a kind of similar related experiment, we also looked at the health of the motor neurons and how long they survived. So here, same basic setup. We take motor neurons that have been grown in a dish, treat some of them with AMX 114 to reduce the levels of calpain 2, and leave some of them untreated as a control. And then we expose them to a different compound, hydrogen peroxide, which is toxic and causes the neurons to die. And we measured essentially how many motor neurons survived in those conditions. And what we see is that the motor neurons that had been pre-treated with AMX 114 and had lower levels of calpain 2 survived much longer than the motor neurons that had not been treated with AMX 114. So on the basis of these experiments, looking at, a few different ways to injure motor neurons, what we see is that treatment with AMX 114 leads to, improvement in the axonal health, less neurofilament light chain kind of being released into the media, as well as improvement in survival of the motor neurons. The next type of experiment that we did is to then move this into an ALS cell model and see kind of, same basic idea. Does treatment with AMX 114 improve the motor neuron health and survival in an ALS cell model? So we did this in conjunction with Sammy Barmata at the University of Michigan. And what Sammy's lab did is essentially create, again, motor neurons in a dish, except this time these motor neurons have a specific ALS-linked mutation. So this is a mutation that we know is linked to and causes ALS in individuals. And then we took those sort of, ALS motor neurons in a petri dish and treated some of them with AMX 114 and some of them were treated with a control. And then we measured both the amounts of neurofilament light chain levels, as well as kind of monitored those neurons over time to see how many of them died. And what we saw is that the motor neurons with the ALS-linked mutation in TDP43 had improvement in the amount of neurofilament light chain that they released, as well as had improvement in survival when they were treated with AMX 114. So all of this data kind of taken together really sort of reinforced our idea that, calpain-2 is kind of this key protein involved in axonal degeneration and is an important early step in ALS disease pathogenesis, and that using this antisense oligonucleotide, AMX 114, can be really protective against those injuries. We then conducted experiments in animals, so both in rats as well as in monkeys, to monitor kind of the safety of AMX 114, and really saw that in general, the animals tolerated the AMX 114 incredibly well, making us even more confident about kind of moving these experiments out of kind of the preclinical stage and into the clinic. So next, I'm going to move into our first in human study design. We call this study LUMINA. So you'll kind of hear me refer to LUMINA throughout the study going forward. So LUMINA is a phase 1 multi-centered randomized placebo-controlled study. It is what's called a multiple ascending dose study, meaning that we are going to be giving individuals who come into the study multiple doses of AMX 114 or placebo during the course of the study. We will also be testing different dose levels, so different amounts of the drug during the study. We are planning to test a total of 4 dose levels of the study drug, either AMX 114 in the placebo, and these will be examined sequentially. So we'll start at the lowest possible dose, and then once we examine that, we'll then move up to a slightly higher dose, and then kind of continue to do that throughout the study. Our main objectives in LUMINA are to evaluate the safety and tolerability of AMX 114 when it is given intrathecally, which means when it's given into the CSF space. And we will be giving a total of 4 doses per dose level. In addition to that, we're going to look at the effects of 114 on the level of key biomarkers, both in the plasma or in the blood, as well as in the cerebrospinal fluid. We will also be measuring some functional measures of ALS disease progression. On the right side of the slide here, I have our key trial entry criteria. So we are looking for adults who are greater than or equal to 18 years of age. They must have a diagnosis of clinically definite or clinically probable ALS, and must have had the time since onset of first symptom of less than 24 months. In addition, because of the way that we're administering the drug, we do want a slow vital capacity, which is a measure of lung function, to be greater than or equal to 75%. And other treatments for ALS are allowed so long as people are on a stable dose for at least 30 days. This is a kind of schematic of our study design. I've already kind of talked about many of the details here, but I'm going to talk about it again here. So As I said, this is kind of a multiple ascending dose study, meaning that we're going to start kind of in the bottom at this very bottom cohort here, cohort 1, where we're giving the lowest dose of the study drug. People who come into the study will be randomized 3-to-1 to either receive AMX 114 or to receive a placebo. Once we look at the safety and tolerability data, as well as the biomarkers from this, we would then dose escalate and open up the next cohort, which would be at a higher dose of the study drug. And then we would kind of continue to serially examine the data and kind of dose escalate from there. We will plan to offer an open label extension for the study once we have had an opportunity to look at the safety and the efficacy data to show that it's, has a positive benefit risk profile to receiving AMX 114. For an individual who comes into this study and decides to participate, this bottom part kind of examines this. So there would be a 4-week screening period. They would then enter into the treatment period, which can last for up to 13 weeks, during which they would receive an intrathecal dose of the drug every 4 weeks for a total of 4 doses. And then there would be an 8-week safety follow-up period at the end. Our endpoints, I've already talked about a little bit here. They are on the slide. So our primary endpoints are really focused on safety. We really want to know, because this is the first time we are giving AMX 114 to people, is it safe and how well tolerated is it? Our secondary endpoints are looking at the concentrations of the study drug AMX 114 in both the plasma as well as in the cerebrospinal fluid. And then we are going to be looking at a number of different biomarkers as well as the ALS disease progression using things like the ALS-FRSR. This really is my last slide. I realize I've been talking quite a bit and excited to get to the Q&A, but I want to highlight that, really throughout the design of this as well as the execution, we here at Amylyx really keep people living with ALS at the center of everything that we do. So as we're kind of thinking through the design of this study, we have incorporated input from all of these different people. So we've talked to a number of people within academia as well as our regulatory agencies. But at the heart of everything is really the people living with ALS. And so we have gathered feedback from the community at multiple times during our study design prior to kind of finalizing our study design plans. Our current status, so we are in the process of initiating LUMINA in Canada as well as in the United States. So we currently have 1 site that is open in Canada. I'm very excited to say that. And we plan to have 3 additional sites in Canada that will be open in the very near future. We are also at the same time working to kind of open additional sites within the United States. And the latest information on the study as well as the sites can be found at our clinicaltrials.gov. If you search for either LUMINA or Amylyx or use this website right here, you can get the most up-to-date information about the study. Great.
Great. Well, thank you very much, Dr. Kett. That was very thorough and great, and I'm glad that you gave some of the preclinical evidence that led you to decide that this was what Amylyx wanted to put forward. And I know I've talked many times about how I know in previous years the company had been going around trying to work really hard to figure out what they might want to try to move forward to clinical development. So it's good to hear how they decided to hang their hat on Calpain 2. So a couple of quick questions. Thank you for acknowledging about the Canadian sites. That was definitely something we want to always ask about. That's great that Canadian sites are being chosen for the phase 1. I did have a question about why an ASO. So, there are inhibitors of Calpain 2 out there. Was it because it's central nervous system through intrathecal route, or is it more specific than, say, like a small molecule?
Yeah, I mean, yes, thank you for the question. And I agree with both of the things that you said. So I'm not going to pick either or, I'm kind of going to go with and. So I think, we think that an ASO is sort of the right way to target Calpain 2 for a few different reasons. So the first is, so Calpain 2 is kind of broadly expressed, not just in the brain and the spinal cord, but in other tissues in the body as well. And so by using an antisense oligonucleotide and delivering it directly into the intrathecal space, space directly into the cerebrospinal fluid, we're able to kind of get that calpain-2 inhibition in the places that we're most interested in. So we're able to kind of, get it up to the brain and the motor cortex. We're also able to get it to the motor neurons that are sitting in the spinal cord, and also kind of, be able to get it into the axons that are going out to the muscles. So we're able to get it to where we want it to go. The second thing that we've seen is from small molecules inhibiting calpain-2, have been kind of looked at and are in the process of being developed. In general, it is hard to inhibit calpain-2 with a small molecule without also inhibiting some of the other calpains. So in particular, there's calpain-1 and calpain-2, and they look very, very similar. And so it's hard to kind of design a small molecule that would only inhibit calpain-2 without inhibiting calpain-1. Whereas with an antisense oligonucleotide, we know that by targeting the mRNA for just calpain-2, we are only knocking down calpain-2 levels. We're not affecting any of the other calpains. We're not affecting the, calpastatin endogenous inhibitor. So those are a few of the different reasons why we thought that the antisense oligonucleotide was a good approach for this specific target.
Great. Thank you. Sounds a very good answer. So actually, we had a number of questions came in, and one kind of related to that is there was a question around what you, and you're a neurologist, so also probably good to be able to answer this. But considering the biggest risk for trial participants in the study or for any kind of ASO trials, particularly first in human ones as well.
Yeah, I mean, I think, obviously with the first in human trial, we're going to be paying very, very close attention to the safety and tolerability of the drug. I think we do studies, both in cell culture dishes as well as in animals to kind of have the most confidence possible that the drug that we're bringing into the clinic is safe. But I think, it is always a little bit, nerve-wracking, I think, to kind of, be in that first in human trial. So I think we have a lot of confidence based on our preclinical data that this drug is safe and will be well tolerated. But I think that we're going to be keeping a very, very close eye on, the safety and monitoring for any adverse effects. That said, I think, in watching, Qalsody and some of the other ASOs that are out there, I think really the thing that I expect to be the most likely side effect are really related to one, the intrathecal administration, as well as two, kind of, when you have to give sort of high doses of antisense oligonucleotides, we know that that has certain effects when given intrathecally. So the intrathecal administration, it's given via lumbar puncture. So, you have to take a small needle, go in at the very bottom of the spine, remove some fluid, and then kind of give the study drug. And lumbar puncture, it is an intervention. It's often associated with things like headache, as well as, kind of back pain. In addition, we know from things like Qalsody that giving high doses of antisense oligonucleotides can have a risk of complications. So I think those are the things that we're going to kind of be particularly watching for. But again, with this being a first in human study, we're going to be paying very close attention to all the safety data.
Sure. And that's also why you do cohorts and you kind of
Exactly.
manage things over a period of time and make sure that, the first ones you do, you're kind of keeping an eye on it before doing more and the multiple ascending dose piece, I think, too. So
That's right.
thank
Yep.
you for that. Yeah, that's great. So just actually, one of your slides talked about the open label extension possibility based on the results that you're kind of getting from the safety tolerability piece. Did it say 8 weeks was the potential of an open label extension or?
No, sorry, it was
Okay.
two separate things. So the 8 weeks really referred to the safety follow-up. So
Okay.
when people come into the trial for an individual, they would come in, they would be screening period, treatment period, and then they would also be monitored, again, back to that safety piece, monitored for an additional 8 weeks to see if there are any kind of long-term side effects from the drug. The open label extension is really separate. That would be, kind of once we have enough data to say, yes, we think there is a reasonable chance of benefit here and, low chance of risk, that's when we would kind of open up an open label extension.
Okay. And to clarify for the
Yep.
person who asked about being on placebo, if you get randomized to placebo, that is what we were referring to right there was that yes, the intention of the company will be to put people on open label on the actual study drug afterwards if it seems like it's the right thing to do for the participants when the study comes because it's so new and novel. And then, just kind of coming back quickly to there was a question around efficacy, and obviously this is a small study and it's hard to get efficacy, but you also have a lot of biomarkers. So
Yep.
neurofilament being one, which is, intriguing right now in terms of what you might be able to tell, and some of your preclinical evidence leads to that. One of the things, so So from that piece, I wouldn't mind if you said something very quickly about it. But then the other piece was target engagement. Is that what the calpain 2 levels? And I saw SBDP 145 might have been something related to
Yes.
that as well,
Yep. Yep.
which is great.
Exactly. So we're going to be looking at biomarkers. And I think what we really want to try to understand is kind of, what are we doing in terms of the biology here? So we're giving AMX 114. Are we knocking down levels of calpain 2? You're right. SBDP 145, which is a mouthful to say, is kind of a proxy for calpain 2 activity. So are we affecting calpain 2 activity? We also know that calpain 2 works directly on things like neurofilament light chain. So are we affecting, neurofilament light chain, which is obviously a biomarker of interest here? And
Yep.
then, even further downstream from that, are we having an effect on kind of like the ALS disease pathogenesis? So that's what we're hoping to understand. Knowing, as you said, that this study is relatively small. We're examining, 48 people over the course of the whole study.
Great. Thank you. Yeah, I love it. The reason why I always ask that question, everyone, is that we learn from the science if we're getting target engagement, so that no matter what happens, we can understand how to move forward to the next step. So really appreciate that answer. And of course, again, the acknowledgement of like we have to still try to understand how these smaller studies can tell us things about efficacy. So that was some of the questions that were in the chat. We're actually already at time. It goes so fast. But thank you so much for the presentation, Dr. Kett. This is great. It's good to see Amylyx back in the ALS space, pushing forward and Canadian sites to be able to participate. So if anyone has any further questions, please do send them to us. If we didn't answer your question effectively, please do email us at research@als.ca. There will be a feedback survey for the first time after this one, so we'll send it out to all participants. We'd love to hear what you think of the Clinical Trials Unboxed webinars. And just looking forward to interacting with you, Dr. Kett, more in the future and Amylyx and seeing how the study progresses. And thank you so much again for being here.
Thank you so much for the invitation. Also happy to kind of, follow up via email or whatever about some of the questions. So Dave, if you want to,
Yep.
send those along, I'm happy to kind of get back to people because I know 30 minutes went by really fast.
Yep. It always does, but we're in and out. And yeah, we'll absolutely make the connections on that. So great. Thank you, everyone. Wishing you a wonderful rest of the day. And we'll see you at the next ALS Clinical Trials Unboxed. See you later.